Don't stop. Don't stop. Don't stop. Don't stop. Don't stop. Don't stop.
Enzymes. Practically all of the numerous and complex biochemical reactions that take place in animals, plants, and microorganisms are regulated by enzymes. These catalytic proteins are efficient and specific, that is, they accelerate the rate of one kind of chemical reaction of one type of compound, and they do so in a far more efficient manner than human-made catalysts. They are controlled by activators and inhibitors that initiate or block reactions. All cells contain enzymes, which usually vary in number and composition, depending on the cell type. An average mammalian cell, for example, is approximately 1/1-billionth, 10^9, the size of a drop of water and generally contains about 3,000 enzymes. The existence of enzymes was established in the middle of the 19th century by scientists studying the process of fermentation. The discovery of the role of enzymes as catalysts followed rapidly. Developments before 1850 included, in 1833, the separation from malt of the enzyme amylase, which converts starch into sugar, and, in 1836, the isolation from the stomach wall of animals of a component of gastric juice that could partially digest food in a test tube, the enzyme pepsin. Enzymes were known for many years as ferments, a term derived from the Latin word for yeast. In 1878 the name enzyme, from the Greek words meaning in yeast, was introduced. Since the late 19th century it has been employed universally. Role of enzymes in metabolism. Some enzymes help to break down large nutrient molecules, such as proteins, fats, and carbohydrates, into smaller molecules. This process occurs during the digestion of foodstuffs in the stomach and intestines of animals. Other enzymes guide the smaller, broken down molecules through the intestinal wall into the bloodstream. Still other enzymes promote the formation of large, complex molecules from the small, simple ones to produce cellular constituents. Enzymes are also responsible for numerous other functions, which include the storage and release of energy, the course of reproduction, the processes of respiration, and vision. They are indispensable to life. Each enzyme is able to promote only one type of chemical reaction. The compounds on which the enzyme acts are called substrates. Enzymes operate in tightly organized metabolic systems called pathways. A seemingly simple biological phenomenon, the contraction of a muscle, for example, or the transmission of a nerve impulse, actually involves a large number of chemical steps in which one or more chemical compounds, substrates, are converted to substances called products. The product of one step in a metabolic pathway serves as the substrate for the succeeding step in the pathway. The role of enzymes in metabolic pathways can be illustrated diagrammatically. The chemical compound represented by A, see diagram below, is converted to product E in a series of enzyme-catalyzed steps, in which intermediate compounds represented by B, C, and D are formed in succession. They act as substrates for enzymes represented by 2, 3, and 4. Compound A may also be converted by another series of steps, some of which are the same as those in the pathway for the formation of E, to products represented by G and H. The letters represent chemical compounds, numbers represent enzymes that catalyze individual reactions. The relative heights represent the thermodynamic energy of the compounds. Example: compound A is more energy-rich than B, B more energy-rich than C. Compounds A, B, etc., change very slowly in the absence of a catalyst but do so rapidly in the presence of catalysts 1, 2, 3, etc. The regulatory role of enzymes in metabolic pathways can be clarified by using a simple analogy, that between the compounds, represented by letters in the diagram, and a series of connected water reservoirs on a slope. Similarly, the enzymes represented by the numbers are analogous to the valves of the reservoir system. The valves control the flow of water in the reservoir, that is, if only valves 1, 2, 3, and 4 are open, the water in A flows only to E, but, If valves 1, 2, 5, and 6 are open, the water in A flows to G. In a similar manner, if enzymes 1, 2, 3, and 4 in the metabolic pathway are active, product E is formed, and, if enzymes 1, 2, 5, and 6 are active, product G is formed. The activity or lack of activity of the enzymes in the pathway therefore determines the fate of compound A, i.e., it either remains unchanged or is converted to one or more products. In addition, if products are formed, the activity of enzymes 3 and 4 relative to that of enzymes 5 and 6 determines the quantity of product E formed compared with product G. Both the flow of water and the activity of enzymes obey the laws of thermodynamics, hence, water in reservoir F cannot flow freely to H by opening valve 7, because water cannot flow uphill. If, however, valves 1, 2, 5, and 7 are open, water flows from F to H, because the energy conserved during the downhill flow of water through valves 1, 2, and 5 is sufficient to allow it to force the water up through valve 7. In a similar way, enzymes in the metabolic pathway cannot convert compound F directly to H unless energy is available. Enzymes are able to utilize energy from energy-conserving reactions in order to catalyze reactions that require energy. During the enzyme-catalyzed oxidation of carbohydrates to carbon dioxide and water, energy is conserved in the form of an energy-rich compound, adenosine triphosphate, ATP. The energy in ATP is utilized during an energy-consuming process such as the enzyme-catalyzed contraction of muscle. Because the needs of cells and organisms vary, not only the activity but also the synthesis of enzymes must be regulated. Example: the enzymes responsible for muscular activity in a leg muscle must be activated and inhibited at appropriate times. Some cells do not need certain enzymes. A liver cell, for example, does not need a muscle enzyme. A bacterium does not need enzymes to metabolize substances that are not present in its growth medium. Some enzymes, therefore, are not formed in certain cells, others are synthesized only when required, and still others are found in all cells. The formation and activity of enzymes are regulated not only by genetic mechanisms but also by organic secretions, hormones, from endocrine glands and by nerve impulses. Small molecules also play an important role, see below enzyme flexibility and allosteric control. If an enzyme is defective in some respect, disease may occur. The enzymes represented by the numbers 1 to 4 in the diagram must function during the conversion of the starting substance A to the product E. If one step is blocked because an enzyme is unable to function, product E may not be formed. If E is necessary for some vital function, disease results. Many inherited diseases and conditions of humans result from a deficiency of one enzyme. Some of these are listed in the table. Albinism, for example, results from an inherited lack of ability to synthesize the enzyme tyrosinase, which catalyzes one step in the pathway by which the pigment for hair and eye color is formed. Enzymes identified with hereditary diseases. Disease name: defective enzyme. Albinism. Tyrosinase. Phenylketonuria. Phenylalanine hydroxylase. Fructosuria. Fructokinase. Methemoglobinemia. Methemoglobin reductase. Galactosemia. Galactose 1-phosphate uridyl transferase. Other functions. Enzymes play an increasingly important role in medicine. The enzyme thrombin is used to promote the healing of wounds. Other enzymes are used to diagnose certain kinds of disease, to cause the remission of some forms of leukemia, a disease of the blood-forming organs, and to counteract unfavorable reactions in people who are allergic to penicillin. The enzyme lysozyme, which destroys cell walls, is used to kill bacteria. Enzymes have also been investigated for their potential to prevent tooth decay and to serve as anticoagulants in the treatment of thrombosis, a disease characterized by the formation of a clot, or plug, in a blood vessel. Enzymes may eventually be used to control enzyme deficiencies and abnormalities resulting from diseases. It might also be noted in passing that enzymes are used in industrial processes involving the preparation of certain chemical compounds and the tanning of leather. They also are valuable in analytical procedures involving the detection of very small quantities of specific substances. Enzymes are necessary in various food-related industries, including cheese making, the brewing of beer, the aging of wine, and the baking of bread. Enzymes also may be used to clean clothes. For some industrial uses of enzymes, see baking. General properties. Classification and nomenclature. The first enzyme name, proposed in 1833, was diastase. 65 years later, French microbiologist and chemist Émile Ducasse suggested that all enzymes be named by adding ase to a root indicative of the nature of the substrate of the enzyme. Although enzymes are no longer named in such a simple manner, with the exception of a few e.g., pepsin, trypsin, chymotrypsin, papain, most enzyme names do end in ase. Any systematic classification of enzymes should be based on a common property or quality that varies sufficiently to be useful as a distinguishing feature. In this regard, three properties of enzymes could serve as a basis for enzyme classification: the exact chemical nature of the enzyme, the chemical nature of the substrate, and the nature of the reaction catalyzed. In addition, although, as indicated above, early attempts at enzyme classification were based on the nature of broad groups of substrates, example enzymes called carbohydrases act on carbohydrates, close functional similarities among enzymes in different groups were often obscured. By general agreement, enzymes now are classified according to their substrates and the nature of the reaction they catalyze. In an attempt to devise a rational system of enzyme nomenclature, two names are given to an enzyme. One, known as the systematic name, is based on logical principles but is often long and awkward. The other, trivial name, is short and generally used but not usually exact or systematic. In the scheme of systematic nomenclature, 6 main groups of enzymatic reactions are recognized. Each catalyzes one reaction type and is subdivided on the basis of detailed definitions of the reaction catalyzed and of the substrate involved in the reaction. Enzymes that catalyze reactions in which hydrogen is transferred belong to the group known as oxidoreductases. Those that catalyze the introduction of the elements of water at a specific site in a molecule are called hydrolyses. The other 4 groups of reactions are the transferases, which catalyze reactions in which substances other than hydrogen are transferred, the lyases, the isomerases, and the ligases. Oxidoreductases and transferases account for about 50% of the approximately 1,000 enzymes recognized thus far. The table lists a few enzymes, their trivial names, their systematic names, and their biological roles. Chemical nature. Little was known about the chemical nature of enzymes until the beginning of the 20th century, although scientists were almost convinced that they were proteins. In 1926 the enzyme urease was the first to be crystallized and clearly identified as a protein. Within the next few years the digestive enzymes pepsin, trypsin, and chymotrypsin were shown to be proteins. Since that time hundreds of enzymes, all of them proteins, have been prepared and characterized by chemical methods. Much of the knowledge of protein chemistry has, in fact, resulted from studies involving enzymes and from attempts to understand their nature and mode of action. Although some enzymes consist of a single chain of the amino acids, i.e., simple organic molecules containing nitrogen, most enzymes are composed of more than one chain. Each chain is called a subunit. Many enzymes have 2, 4, or 6 subunits, and some consist of as many as 12 to 60 subunits. In many cases the subunits have identical structures, in others, however, several different types of subunit chains are involved. With the exception of proteins that act as structural elements, most of the proteins in physiologically active tissues such as kidney and liver are enzymes. Regardless of the exact amount of enzymatic protein in an organism, it is clear that hundreds of different enzymes must be present in each tissue to account for the myriad reactions composing metabolism. Cofactors. Although some enzymes consist only of protein, many are complex proteins, i.e., they have a protein component and a so-called cofactor. A complete enzyme is called a holoenzyme. If the cofactor is removed, the protein, no longer enzymatically active, is called the apoenzyme. A cofactor may be a metal such as iron, copper, or magnesium, a moderately sized organic molecule called a prosthetic group, or a special type of substrate molecule known as a coenzyme. The cofactor may aid in the catalytic function of an enzyme, as do metals and prosthetic groups, or take part in the enzymatic reaction, as do coenzymes. B-vitamin coenzymes in metabolism. Functions of B-vitamin coenzymes in metabolism. A coenzyme serves as a type of substrate in certain enzymatic reactions and thus reacts in the exact proportions, i.e., stoichiometrically, required for reaction, rather than in catalytic quantities. A coenzyme may, for example, assume the role of a hydrogen acceptor, as does nicotinamide adenine dinucleotide, NAD, which accepts hydrogen from the substrate, or a chemical group donor, as does adenosine triphosphate, ATP, which donates phosphoric acid to the substrate. After ATP has donated a phosphoric acid molecule to the substrate, the phosphoric acid can be reacquired in a second stoichiometric reaction catalyzed by a second enzyme. The catalytic nature of a coenzyme is apparent only when it couples the activities of two enzymes in this way. Coenzymes thus are the links, or shuttles, in metabolic pathways that enable substances, e.g., hydrogen, phosphoric acid, to be exchanged. The nature of enzyme-catalyzed reactions. The nature of catalysis. In a chemical reaction, for example, one in which substance A is converted into product B, a point of equilibrium eventually is reached at which no further chemical change occurs, i.e., the rate of conversion of A to B equals the rate of conversion of B to A. The so-called thermodynamic equilibrium constant expresses this chemical equilibrium. A catalyst may be defined as a substance that accelerates a chemical reaction but is not consumed in the process. The amount of catalyst has no relationship to the quantity of substance altered. Very small amounts of enzymes are very efficient catalysts. Because the presence of an enzyme accelerates the rate of conversion of a compound to a product, it accelerates the approach to equilibrium. It does not, however, influence the equilibrium point attained. The molecules in the watery medium of the cell are in constant thermal motion, but, because they are more or less stable compounds, they would react only occasionally to form products in the absence of enzymes. There exists an energy barrier to the reaction of a molecule. The energy required to overcome the barrier to reaction is called the energy of activation. A reaction proceeds to equilibrium only if the molecules have sufficient energy of activation to form an activated complex, from which products can be derived. Enzymes greatly increase the chances for reactions by their ability to make large numbers of specific molecules more reactive, i.e., unstable, by forming intermediate compounds with them. The unstable intermediates quickly break down to form stable products, and the enzymes, unchanged by the reaction, are able to catalyze the formation of additional products. The role of the active site. That the compound on which an enzyme acts, substrate, must combine in some way with it before catalysis can proceed is an old idea, now supported by much experimental evidence. The combination of substrate molecules with enzymes involves collisions between the two. Enzymes are large molecules, the molecular weights of which, based on the weight of a hydrogen atom as one, range from several thousand to several million. The substrates on which enzymes act usually have molecular weights of several hundred. Because of the difference in size between the two, only a fraction of the enzyme is in contact with the substrate. The region of contact is called the active site. Usually, each subunit of an enzyme has one active site capable of binding substrate. The characteristics of an enzyme derive from the sequence of amino acids, which determine the shape of the enzyme, i.e., the structure of the active site, and hence the specificity of the enzyme. The forces that attract the substrate to the surface of an enzyme may be of a physical or a chemical nature. Electrostatic bonds may occur between oppositely charged groups. The circles containing plus and minus signs on the enzyme are attracted to their opposites in the substrate molecule. Such electrostatic bonds can occur with groups that are completely positively or negatively charged, i.e., ionic groups, or with groups that are partially charged, i.e., dipoles. The attractive forces between substrate and enzyme may also involve so-called hydrophobic bonds, in which the oily, or hydrocarbon, portions of the enzyme, represented by H-labeled circles, and the substrate are forced together in the same way as oil droplets tend to coalesce in water. Enzyme, active site. The role of the active site in the lock and key fit of a substrate, the key, to an enzyme, the lock. Modifications in the structure of the amino acids at or near the active site usually affect the enzyme's activity, because these amino acids are intimately involved in the fit and attraction of the substrate to the enzyme surface. The characteristics of the amino acids near the active site determine whether or not a substrate molecule will fit into the site. A molecule that is too bulky in the wrong places cannot fit into the active site and thus cannot react with the enzyme. In a similar manner, a molecule lacking essential attractive forces or the appropriately charged regions might not be bound to the enzyme. On the other hand, a molecule with a bulky group at a position such that it does not interfere with the binding of the molecule to the enzyme or with the function of the active site is able to serve as a substrate for the enzyme. The idea of a fit between substrate and enzyme, called the key-lock hypothesis, was proposed by German chemist Emil Fischer in 1899 and explains one of the most important features of enzymes, their specificity. In most of the enzymes studied thus far, a cleft, or indentation, into which the substrate fits is found at the active site. The specificity of enzymes. Since the substrate must fit into the active site of the enzyme before catalysis can occur, only properly designed molecules can serve as substrates for a specific enzyme. In many cases, an enzyme will react with only one naturally occurring molecule. Two oxidoreductase enzymes will serve to illustrate the principle of enzyme specificity. 1. Alcohol dehydrogenase, acts on alcohol, the other, lactic dehydrogenase, on lactic acid. The activities of the two, even though both are oxidoreductase enzymes, are not interchangeable, i.e., alcohol dehydrogenase will not catalyze a reaction involving lactic acid or vice versa, because the structure of each substrate differs sufficiently to prevent its fitting into the active site of the alternative enzyme. Enzyme specificity is essential because it keeps separate the many pathways, involving hundreds of enzymes, that function during metabolism. Not all enzymes are highly specific. Digestive enzymes such as pepsin and chymotrypsin, for example, are able to act on almost any protein, as they must if they are to act upon the varied types of proteins consumed as food. On the other hand, thrombin, which reacts only with the protein fibrinogen, is part of a very delicate blood clotting mechanism and thus must act only on one compound in order to maintain the proper functioning of the system. When enzymes were first studied, it was thought that most of them were absolutely specific, that they would react with only one compound. In most cases, however, a molecule other than the natural substrate can be synthesized in the laboratory. It is enough like the natural substrate to react with the enzyme. Use of these synthetic substrates has been valuable in understanding enzymatic action. It must be remembered, however, that, in the living cell, many enzymes are absolutely specific for the compounds found there. All enzymes isolated thus far are specific for the type of chemical reaction they catalyze, i.e., oxidoreductases do not catalyze hydrolase reactions, and hydrolases do not catalyze reactions involving oxidation and reduction. An enzyme therefore catalyzes a specific chemical reaction but may be able to do so on several similar compounds. The mechanism of enzymatic action. An enzyme attracts substrates to its active site, catalyzes the chemical reaction by which products are formed, and then allows the products to dissociate, separate from the enzyme surface. The combination formed by an enzyme and its substrates is called the enzyme-substrate complex. When two substrates and one enzyme are involved, the complex is called a ternary complex. One substrate and one enzyme are called a binary complex. The substrates are attracted to the active site by electrostatic and hydrophobic forces, which are called non-covalent bonds because they are physical attractions and not chemical bonds. Mechanisms of enzymatic action. As an example, assume two substrates, S1 and S2, bind to the active site of the enzyme during step 1 and react to form products, P1 and P2, during step 2. The products dissociate from the enzyme surface in step 3, releasing the enzyme. The enzyme, unchanged by the reaction, is able to react with additional substrate molecules in this manner many times per second to form products. The step in which the actual chemical transformation occurs is of great interest, and, although much is known about it, it is not yet fully understood. In general, there are two types of enzymatic mechanisms, one in which a so-called covalent intermediate forms and one in which none forms. In the mechanism by which a covalent intermediate, i.e., an intermediate with a chemical bond between substrate and enzyme forms, one substrate, Bx, for example, reacts with the group N on the enzyme surface to form an enzyme B intermediate compound. The intermediate compound then reacts with the second substrate, Y, to form the products By and X. Many enzymes catalyze reactions by this type of mechanism. Acetylcholinesterase is used as a specific example in the sequence described below. The two substrates, S1 and S2, for acetylcholinesterase are acetylcholine, i.e., Bx, and water, Y. After acetylcholine, Bx, binds to the enzyme surface, a chemical bond forms between the acetyl moiety, B, of acetylcholine and the group N, part of the amino acid serine, on the enzyme surface. The result of the formation of this bond, called an acyl-serine bond, is one product, choline, X, and the enzyme B intermediate compound, an acetyl-enzyme complex. The water molecule, Y, then reacts with the acyl-serine bond to form the second product, acetic acid, By, which dissociates from the enzyme. Acetylcholinesterase is regenerated and is again able to react with another molecule of acetylcholine. This kind of reaction, involving the formation of an intermediate compound on the enzyme surface, is generally called a double displacement reaction. Sucrose phosphorylase acts in a similar way. The substrate for sucrose phosphorylase is sucrose, or glucosylfructose, Bx, and the group N on the enzyme surface is a chemical group called a carboxyl group, COOH. The enzyme B intermediate, a glucosyl-carboxyl compound, reacts with phosphate, Y, to form glucosyl phosphate, By. The other product, X, is fructose. In double displacement reactions, the covalent intermediate between enzyme and substrate apparently influences the reaction to proceed more rapidly. Because the enzyme is unaltered at the end of the reaction, it functions as a true catalyst, even though it is temporarily altered during the enzymatic process. Although many enzymes form a covalent intermediate, the mechanism is not essential for catalysis. One substrate, Y, reacts directly with the second substrate, XB, in a so-called single displacement reaction. The B moiety, which is transformed in the chemical reaction, is involved in only one reaction and does not form a bond with a group on the enzyme surface. The enzyme maltose phosphorylase, for example, directly affects the bonds of the substrates, Bx and X, which, in this case, are maltose, glucosyl-glucose, and phosphate, to form the products, glucose, X, and glucosyl phosphate, By. Covalent intermediates between part of a substrate and an enzyme occur in many enzymatic reactions, and various amino acids serine, cysteine, lysine, and glutamic acid are involved. The rate of enzymatic reactions. The Michaelis-Menten hypothesis. If the velocity of an enzymatic reaction is represented graphically as a function of the substrate concentration, S, the curve obtained in most cases is a hyperbola. The mathematical expression of this curve, shown in the equation below, was developed in 1912-13 by German biochemists Leonor Michaelis and Maud Leonora Menten. In the equation, Vm is the maximal velocity of the reaction, and Km is called the Michaelis constant, diagram of enzyme action. The shape of the curve is a logical consequence of the active site concept, i.e., the curve flattens at the maximum velocity, Vm, which occurs when all the active sites of the enzyme are filled with substrate. The fact that the velocity approaches a maximum at high substrate concentrations provides support for the assumption that an intermediate enzyme, substrate complex forms. At the point of half the maximum velocity, the substrate concentration in moles per liter, m, is equal to the Michaelis constant, which is a rough measure of the affinity of the substrate molecule for the surface of the enzyme. Km values usually vary from about 10^8 to 10^2 m, and Vm from 105 to 109 molecules of product formed per molecule of enzyme per second. The value for Vm is referred to as the turnover number when expressed as moles of product formed per mole of enzyme per minute. The binding of molecules that inhibit or activate the protein surface usually results in similar types. Enzymes are more efficient than human-made catalysts operating under the same conditions. Because many enzymes with different specificities occur in a cell, adequate space exists only for a few enzyme molecules catalyzing one specific reaction. Each enzyme, therefore, must be very efficient. One molecule of the enzyme catalase, for example, can produce 1012 molecules of oxygen per second. The catalytic groups at the active site of an enzyme act 106 to 109 times more effectively than do analogous groups in a non-enzymatic reaction. The reason for the great efficiency of enzymes is not completely understood. It results in part from the precise positioning of the substrates and the catalytic groups at the active site, which serves to increase the probability of collision between the reacting atoms. In addition, the environment at the active site may be favorable for reaction, that is, acidic and basic groups may act together more effectively there, or some strain may be induced in the substrate molecules so that their bonds are broken more easily, or the orientation of the reacting substrates may be optimal at the enzyme surface. The theories that have been formulated to account for the high catalytic efficiency of enzymes, although reasonable, still remain to be proved. Inhibition of enzymes. Some molecules very similar to the substrate for an enzyme may be bound to the active site but be unable to react. Such molecules cover the active site and thus prevent the binding of the actual substrate to the site. This inhibition of enzyme action is of a competitive nature, because the inhibitor molecule actually competes with the substrate for the active site. The inhibitor sulfanilamide, for example, is similar enough to a substrate, p-aminobenzoic acid, of an enzyme involved in the metabolism of folic acid that it binds to the enzyme but cannot react. It covers the active site and prevents the binding of p-aminobenzoic acid. This enzyme is essential in certain disease-causing bacteria but is not essential to humans. Large amounts of sulfanilamide therefore kill the microorganism but do not harm humans. Inhibitors such as sulfanilamide are called antimetabolites. Sulfanilamide and similar compounds that kill a pathogen without harming its host are widely used in chemotherapy. Some inhibitors prevent, or block, enzymatic action by reacting with groups at the active site. The nerve gas diisopropyl fluorophosphate, for example, reacts with the serine at the active site of acetylcholinesterase to form a covalent bond. The nerve gas molecule involved in bond formation prevents the active site from binding the substrate, acetylcholine, thereby blocking catalysis and nerve action. Iodoacetic acid similarly blocks a key enzyme in muscle action by forming a bulky group on the amino acid cysteine, which is found at the enzyme's active site. This process is called irreversible inhibition. Some inhibitors modify amino acids other than those at the active site, resulting in loss of enzymatic activity. The inhibitor causes changes in the shape of the active site. Some amino acids other than those at the active site, however, can be modified without affecting the structure of the active site. In these cases, enzymatic action is not affected. Such chemical changes parallel natural mutations. Inherited diseases frequently result from a change in an amino acid at the active site of an enzyme, thus making the enzyme defective. In some cases, an amino acid change alters the shape of the active site to the extent that it can no longer react. Such diseases are usually fatal. In others, however, a partially defective enzyme is formed, and an individual may be very sick but able to live. Effects of temperature. Enzymes function most efficiently within a physiological temperature range. Since enzymes are protein molecules, they can be destroyed by high temperatures. An example of such destruction, called protein denaturation, is the curdling of milk when it is boiled. Increasing temperature has two effects on an enzyme. First, the velocity of the reaction increases somewhat, because the rate of chemical reactions tends to increase with temperature, and, second, the enzyme is increasingly denatured. Increasing temperature thus increases the metabolic rate only within a limited range. If the temperature becomes too high, enzyme denaturation destroys life. Low temperatures also change the shapes of enzymes. With enzymes that are cold sensitive, the change causes loss of activity. Both excessive cold and heat are therefore damaging to enzymes. The degree of acidity or basicity of a solution, which is expressed as pH, also affects enzymes. As the acidity of a solution changes, i.e., the pH is altered, a point of optimum acidity occurs, at which the enzyme acts most efficiently. Although this pH optimum varies with temperature and is influenced by other constituents of the solution containing the enzyme, it is a characteristic property of enzymes. Because enzymes are sensitive to changes in acidity, most living systems are highly buffered, i.e., they have mechanisms that enable them to maintain a constant acidity. This acidity level, or pH, is about 7 in most organisms. Some bacteria function under moderately acidic or basic conditions, and the digestive enzyme pepsin acts in the acid milieu of the stomach. Enzyme flexibility and allosteric control. The induced fit theory. The key, lock hypothesis, see above. The nature of enzyme-catalyzed reactions, does not fully account for enzymatic action, i.e., certain properties of enzymes cannot be accounted for by the simple relationship between enzyme and substrate proposed by the key, lock hypothesis. A theory called the induced fit theory retains the key, lock idea of a fit of the substrate at the active site, but postulates in addition that the substrate must do more than simply fit into the already preformed shape of an active site. Rather, the theory states, the binding of the substrate to the enzyme must cause a change in the shape of the enzyme that results in the proper alignment of the catalytic groups on its surface. This concept has been likened to the fit of a hand in a glove, the hand, substrate, inducing a change in the shape of the glove, enzyme. Although some enzymes appear to function according to the older key, lock hypothesis, most apparently function according to the induced fit theory. Typically, the substrate approaches the enzyme surface and induces a change in its shape that results in the correct alignment of the catalytic groups. In the case of the digestive enzyme carboxypeptidase, for example, the binding of the substrate causes a tyrosine molecule at the active site to move by as much as 15 angstroms. The catalytic groups at the active site react with the substrate to form products. The products separate from the enzyme surface, and the enzyme is able to repeat the sequence. Non-substrate molecules that are too bulky or too small alter the shape of the enzyme so that a misalignment of catalytic groups occurs. Such molecules are not able to react even if they are attracted to the active site. Diagram of induced fit binding of a substrate to an enzyme surface. The induced fit theory explains a number of anomalous properties of enzymes. An example is non-competitive inhibition, in which a compound inhibits the reaction of an enzyme but does not prevent the binding of the substrate. In this case, the inhibitor compound attracts the binding group so that the catalytic group is too far away from the substrate to react. The site at which the inhibitor binds to the enzyme is not the active site and is called an allosteric site. The inhibitor changes the shape of the active site to prevent catalysis without preventing binding of the substrate. An inhibitor also can distort the active site by affecting the essential binding group. As a result, the enzyme can no longer attract the substrate. A so-called activator molecule affects the active site so that a non-substrate molecule is properly aligned and hence can react with the enzyme. Such activators can affect both binding and catalytic groups at the active site. Enzyme flexibility is extremely important because it provides a mechanism for regulating enzymatic activity. The orientation at the active site can be disrupted by the binding of an inhibitor at a site other than the active site. Moreover, the enzyme can be activated by molecules that induce a proper alignment of the active site for a substrate that alone cannot induce this alignment. As mentioned above, the sites that bind inhibitors and activators are called allosteric sites to distinguish them from active sites. Allosteric sites are in fact regulatory sites able to activate or inhibit enzymatic activity by influencing the shape of the enzyme. When the activator or inhibitor dissociates from the enzyme, it returns to its normal shape. Thus, the flexibility of the protein structure allows the operation of a simple, reversible control system similar to a thermostat. Types of allosteric control. Allosteric control can operate in many ways. Two examples serve to illustrate some general effects. A pathway consisting of 10 enzymes is involved in the synthesis of the amino acid histidine. When a cell contains enough histidine, synthesis stops an appropriate economy move by the cell. Synthesis is stopped by the inhibition of the first enzyme in the pathway by the product, histidine. The inhibition of an enzyme by a product is called feedback inhibition, i.e., a product many steps removed from an initial enzyme blocks its action. Feedback inhibition occurs in many pathways in all living things. Allosteric control can also be achieved by activators. The hormone adrenaline, epinephrine, acts in this way. When energy is needed, adrenaline is released and activates, by allosteric activation, the enzyme adenyl cyclase. This enzyme catalyzes a reaction in which the compound cyclic adenosine monophosphate, cyclic AMP, is formed from ADP. Cyclic AMP in turn acts as an allosteric activator of enzymes that speed the metabolism of carbohydrate to produce energy. This type of allosteric regulation also is widespread in biological systems. Thus, a combination of allosteric activation and inhibition allows the production of energy or materials when they are needed and shuts off production when the supply is adequate. Allosteric control is a rapid method of regulating products continuously needed by living things. Yet some cells have no need for certain enzymes, and it would be wasteful for the cell to synthesize them. In this case, certain molecules, called repressors, prevent the synthesis of unneeded enzymes. The repressors are proteins that bind to DNA and prevent the first step in the process resulting in protein synthesis. If certain metabolites are added to cells that need an enzyme, enzyme synthesis occurs, i.e., it is induced. Addition of galactose to a growth medium containing Escherichia coli bacteria, for example, induces the synthesis of the enzyme beta-galactosidase. The bacteria thus can synthesize this galactose metabolizing enzyme when it is needed and prevent its synthesis when it is not. The way in which the synthesis of enzymes is induced or repressed in mammalian systems is less understood but is believed to be similar. Different types of cells in complex organisms have different enzymes, even though they have the same DNA content. The enzymes actually synthesized are the ones needed in a specific cell and vary not only for different types of cells, e.g., nerve, muscle, eye, and skin cells, but also for different species. In an enzyme consisting of several subunits, or chains, alteration in the shape of one chain as a result of the influence either of a substrate molecule or of allosteric inhibitors or activators may change the shape of a neighboring chain. As a result, the binding of a second molecule of substrate occurs in a different way from the binding of the first, and the third is different from the second. This phenomenon, called cooperativity, is characteristic of allosteric enzymes. Cooperativity is reflected by a sigmoid curve, as compared to the hyperbolic curve of Michaelis, Menten. An enzyme of several subunits that exhibits cooperativity is far more sensitive to control mechanisms than is an enzyme of one subunit and hence one active site. The first example of cooperativity was observed in hemoglobin, which is not an enzyme but behaves like one in many ways. The absorption of oxygen in the lungs and its deposition in the tissues is far more efficient because the subunits of hemoglobin show positive cooperativity, so called because the first molecule of substrate makes it easier for the next to bind. Negative cooperativity, in which the binding of one molecule makes it less easy for the next to bind, also occurs in living things. Negative cooperativity makes an enzyme less sensitive to fluctuations in concentrations of metabolites and may be important for enzymes that must be present in the cell at relatively constant levels of activity. Some enzymes are closely associated aggregates of several enzyme units. The pyruvate dehydrogenase system, for example, contains 5 different enzymes, has a total molecular weight of 4 million, and consists of 4 different types of chains. Apparently, the enzymes in cells may be organized by forming complex units, by being absorbed on a cell wall, or by being isolated by membranes in special compartments. Since a pathway involves the stepwise modification of chemical compounds, aggregations of the enzymes in a given pathway facilitate their function in a manner similar to an industrial assembly line.